Good afternoon. Welcome to the educational session for CAR T cells. The title of this session is CAR T cell design and function improvements. We've had incredible success with CAR T cells that culminated in 3 FDA approvals for 2 therapies for 2 indications in the last 8 months. But it's time now to go from the bedside back to the bench to learn more about these molecules and formulate improvements to their design and to their function. So the speakers that we have lined up for this session, I believe will do that for us. The first speaker is Maksim Mamonkin. Did I pronounce your name correctly, Max? Close. From the Baylor College of Medicine, who will talk to us about structure-function relationships in chimeric antigen receptors.
Thanks a lot, Avery. Thanks to the committee for inviting me here. So I'll talk about the main structural components of a CAR and how they affect the antigen-driven signaling, and also in the context of tonic CAR signaling in T cells. So many of you here in the room are already familiar with CARs. They are usually depicted like this. They have an extracellular domain that is derived from an antibody, antigen recognition domain, in which both heavy and light variable regions are stitched together via a flexible linker in a single chain variable fragment that is connected through a spacer of some sort in a transmembrane domain with the signaling endodomains inside that provide signal 1 for this T cell to degranulate and kill the tumor cell, and signal 2 to provide additional motivation for proliferation, survival, and so forth. And so I'll be talking about all these domains one by one and review them quite concisely, if possible. So most of the time, the antigen binding domain is derived from an antibody in mice or human antibody, but it doesn't have to be so. One can use also antibody or antibody-like sequences from fish or from camels or llamas. They naturally do not have light chain, and so the entire antigen recognition is actually programmed in a single heavy chain domain. Likewise, one can use the human type 2 fibronectin repeats or direct ankyrin repeats to program and to engineer them to recognize your target specifically, and these are even smaller than a VH region. And obviously one can use a natural ligand, if available, to bind the surface molecule that you're targeting, and examples are shown there on the right. Selecting the right binder and the right epitope is critical sometimes for T cells to exert optimal function, as reported in this study by Remo Sorrentis' group, who designed the CD28.2 CAR. And they show that selecting a CAR that binds the membrane proximal region is actually superior to the binders that bind the membrane distal region, indicating that probably there's an optimal space between a T cell and a target cell to form a productive immunologic synapse. Likewise, your CAR may bind antigens, but we only exert the effector function against cells that have very high density, which exceeds the normal level that is expressed in tumors, as exemplified here in the report from Crystal Mackel's group. You can see that the full effector function is exerted against targets with very high expression of ALK, and this exceeds the normal expression range. So one has to be mindful of this when designing a CAR and selecting the optimal binder. CARs can be engineered to bind 2 or more antigens, such as shown here, a tandem CAR or a TAN CAR. In this case, the CAR may have 2 single chain FVs or 2 natural ligands and so forth that allow for dual targeting and offset antigen escape. So you can implement Boolean OR gates, Boolean OR logic. We'll hear more about Boolean logics later in the session from Cole. And when both antigens are bound, this actually promotes formation of very stable synapses and promotes a robust cytotoxic activity. And an example of this is shown on the right, a CAR that binds both IL-13 receptor alpha-2 and HER2. It was initially developed by Nabil Ahmed in our center, followed by others. Another way to implement targeting multiple antigens by a single CAR is to use soluble adapters that would bind individually different antigens, and will have a conserved domain that can be bound by a CAR, and therefore the CAR will recognize these antigens indirectly through a soluble adapter. And this also allows an additional layer of controlling T cell function, because the CARs are only active when these soluble adapters are present. And examples are shown here on the right, on the bottom, I'm sorry. Moving on to the spacer. The spacer region is required to connect antigen binding domains with the signaling domains, and there's a bunch of spacer regions currently used in the clinic and in the commercial development. Some of them are derived from FC spacers, some of them are CD8 or CD28 derived, and they're here shown ranked by their relative length. And the spacers play an important role, and sometimes they are required to facilitate optimal binding of the antigen in certain settings, and selecting the wrong spacer may actually jeopardize the ability of a CAR to properly recognize the target. One can also integrate epitopes for antibodies to facilitate either detection in vivo or selection in vitro of the CAR T cells. There were reports that long FC spacer can interact with other immune cells by means of the CH2 domain. It binds to the FC receptor and is actually detrimental to CAR T cell function, and once you remove it in the intermediate spacer, which is second from the left, this activity is eliminated. Finally, we and others often see that the selection of a spacer or the transmembrane domain actually can modulate the expression level of your CAR on the cell surface. And this is sometimes a good or a bad thing, depending on the context, but there are certain signaling on the domains that are requiring, for example, ICOS that can only be expressed with the ICOS transmembrane domain, and it's very picky about it. So one needs to really do due diligence when selecting the optimal spacer. One feature that's often overlooked in spacers is that they're capable of forming dimers. All of the spacers that are shown here actually can form the disulfide bonds between each other, and so what we see when we run protein gels is that the CARs are actually migrating as dimers or trimers, so they are already expressed at the cell surface as not as monomers. And once you reduce them with beta-mercaptophenol, they all migrate as monomers. Moving on to the signaling endodomains, there's a lot of research past and ongoing, so I'll try to summarize it very quickly. The first generation CARs only have the zeta chain signaling, which is sufficient to cause degranulation of T cells, and it's mimicking TCR signaling in this case, or it can be substituted by other item containing domains like FCR gamma or DAP12. But it is insufficient to enable cells to proliferate and sustain continuous expansion and long-term anti-tumor activity. And therefore, the first CAR, that's called the second generation CAR, was initially published by Michel Sutherland group in 2002, where they showed that incorporation of CD28 signaling, which is a normal co-stimulation signal 2 for T cells, is actually beneficial for these CAR T cells, and it promotes enhanced proliferation and production of cytokines like IL-2, and ultimately leads to very robust effector functions. The CD28 elicits multiple signaling pathways, most commonly known the PI3 kinase and also ITK, and these additional pathways are actually helping T cells to fulfill their full potential. 2 years later, in 2004, Dario Campana in St. Jude's showed that the incorporation of TNF receptor superfamily domain 41BB was actually also beneficial to the T cells. It provided enhanced survival. And 41BB signals differently, signals through TRAC molecules and activates an FKB, which is shown to be critical to maintain survival of the cells. And later studies shown that 41BB signaling can promote central memory development and endow cells with long persistence, and also switch their metabolism. And we will hear more about CAR T cell metabolism from the next speaker. Likewise, later studies show that one can use other domains from the same superfamilies like OX40 and CD27 to substitute for BB, and that leads to slightly different effects, but still the signaling largely is conserved. Likewise, ICOS is another member in the O5G superfamily. It primarily activates PI3 kinase and was shown to be beneficial in several settings. Finally, one can combine TNF receptor and IG superfamily domains into one. It's called third generation CAR, and the first one was published back in 2004 by Malcolm Brenner and Martin Poulet, where they combined CD28 and LUX40, and shown that this actually this construct promoted very high secretion of cytokines and improved proliferation. And one also can combine 41BB domain with either CD28, which was published by Michel Sutherland group back in 2010, or ICOS in a recent report by Carl June group at Penn. It was shown to be actually eliciting quite beneficial signaling and promote persistence and survival of the CAR T cells. So how do CARs signal? We have very limited understanding how they actually are signaling, but the conventional model says that it is promoted by binding the antigen and bringing together CAR molecules that form a cluster and leads to cross-linking of signaling domains and activation of signaling, much like it's happening for the TCR in TCR microclusters. And so CARs also possess a few other features that resemble TCR, such as that immediately after engaging the antigen, CAR is often downregulated quite rapidly, as you can see here in the report from Crystal Mackel's group, where they co-cultured T cells expressing CD19 CAR that is fused with a fluorescent protein FP. You can see that upon culturing with the CD19 positive targets, a lot of cells became kind of quasi-CD19 negative, but once they were replated in CD19 negative cells, they re-expressed the CAR. And report that came out the same year from Michel Sutherland's group showed that it doesn't only happen on the protein level, but also at the mRNA level. The CARs are downregulated quite rapidly immediately after encountering the antigen, and then they gradually expression restored back. And the magnitude of and the kinetics of this re-expression really depends on the choice of the promoter that is driving the CAR expression. Is antigen-induced signaling the only way by which CARs can signal? So the answer is no. If you express the CARs at a very high level in T cells and after exceeding certain antigen density, CAR density threshold in the membrane, the CAR molecules start kind of bumping into each other and start forming these clusters spontaneously in the absence of any external antigens. And that's what's called ligand-independent tonic signaling. This is a constitutive signaling that happens in CAR T cells that is driven by this spontaneous clustering. And you can actually see it if you look under the microscope, as shown here in Chris Michael's paper a few years ago, that the CAR, for example, C19 CAR, shown is expressed quite nicely around the cell, and the distribution is more or less even, whereas GD2 CAR, which is much more prone to this tonic signaling, forms this classic punctate distribution resembling these clusters that I was talking about. And interestingly, these clusters are also formed in the CARs that do not have active signaling going on, where the signaling domains are actually mutated, indicating that this clustering is not a result of the signaling, but rather something that probably precedes it. So what are the factors that regulate it? There are two major factors that drive tonic signaling in T cells. First is the biophysical properties, the capacity of the CAR molecules to stick together, to adhere to each other. And this depends a lot on the choice of the scFv, on the structure, on the backbone, and so on and so forth. And the second important factor is the level of expression. Even if your CAR does not produce a lot of tonic signaling normally, you can overexpress it to a point where this density is so high they will be forced to interact with each other and form the clusters. So let me show you a few examples how the CAR structure can actually affect the magnitude of CAR signaling. So the first example I already shown you where it depending on the scFv, in this case the GD2 binder, 0F14G2A, is much more prone to this spontaneous clustering than the CD19 with FMC63, scFv as illustrated in that graph. Likewise, the choice of the spacer and hinge region may affect the propensity of CARs to stick together. And it was studied by Juan Vera and Ann Lin in our center, where this is showing the expansion of CAR T cells after initial stimulation. It's often used as a readout of tonic signaling, so the more the cells expand autonomously after initial stimulation in the absence of other stimulations, the more additional tonic signaling they are receiving. And so here you can see that the cells that have the CARs with a long spacer expanded more than the control T cells, indicating that these CARs are more prone to tonic signaling with a CD28 at least. Likewise, a study from the Penn's group a few years ago showed that the CD28 endodomain is actually more prone to tonic signaling compared to 4-1BB and ICOS. As you can see here, again, the cells expanded much longer when they had a CD28 endodomain in a CAR. But despite the fact that these cells seem to have additional increased expansion in vitro, the conclusion from all these three studies is that the presence of tonic signaling invariably impairs the anti-tumor function of T cells in vivo. And this has something to do with precluding the optimal expansion of the cells and making them unable to persist in the long term, indicating that constant buzzing of the CAR is not a good thing for T cells. And so what is the mechanism that can be attributed to it? We've been studying CARs that tonically signaling with different magnitude, and what we consistently see is when we put a CAR that has a CD28 endodomain and has a very mild tonic signaling, for example, is that these T cells start losing the CCR7 positive population, that is central memory naive-like cells, which are known to be the cells with very high proliferative potential in vivo. And so when this signaling is going on, these populations are actually wiped out gradually. Interestingly, when we replace it with a 4-1BB, this process seems to be a little bit attenuated, and we see more central memory formation, which is consistent with the property of 4-1BB to actually promote central memory in T cells. And in general, the CCR7 positive cells are retained in a little more, and is quantified here. So you can see that the presence of a CD28 endodomains dramatically reduce the frequency of cells that are capable of long expansion in vivo. Another mechanism that was shown by Crystal Mackel's group a few years ago demonstrates that if you overexpress a CAR at a very, very high level, its cells start upregulating activation markers / exhaustion markers like PD-1 and TIM3 and LAG3. So T cells really start thinking that they're receiving real antigen stimulation instead of that tonic antigen independent stimulation, and so that results in the upregulation of those exhaustion markers in T cells and developing of the exhaustion phenotype in the long run. And once you replace the CD28 with a 4-1BB, actually that seems to alleviate the effect quite a bit, and the upregulation of these exhaustion markers is reduced, and the cells look a bit cleaner, as you can see in the pie charts on the right. And probably both of these mechanisms are contributing to the superior function of the 4-1BB CARs in vivo, as demonstrated here, and also leads to a better expansion of these CAR T cells in the blood. I'm going to switch gears and illustrate how the level of expression can affect the magnitude of tonic signaling. And so we modeled this in our recent publication when we used a gamma retroviral system that drives really high expression level of a CAR, in this case containing 4-1BB endodomain. And as a control, we used a construct in which the expression of the same CAR was attenuated by an iris element right upstream. And so what we found is that actually quite the opposite of what is seen with the 28 zeta CAR. The cells that received a lot of tonic signaling from 4-1BB did not expand well, and they actually had quite increased apoptosis and signs of activation-induced cell death. And what we observed ultimately is that this very interesting interplay between the tonic signaling and the expression vector that is driving the CAR expression. In this particular case, continuous signaling from the 4-1BB activates an FKB, which is a great pathway to have. It really promotes survival of T cells. But in this particular case, it also activates expression of the CAR by activating an LTR promoter, and this amplifies production of the CAR and amplifies the tonic signaling from the CAR as a whole. And this leads to overstimulation of the cells and upregulation of FAS and TRAIL and ICAM1 and ultimately activation-induced cell death. So one has to be very mindful of this feedback loop when using 4-1BB CARs between the retroviral vectors. Another facet of toxicity from continuous CAR signaling is actually suppressed responsiveness through the TCR. This happens when we express a highly tonically signaling CAR in multivirus-specific T cells here, and then ask them to produce interferon gamma in response to the specific peptide stimulation. You can see that in both donors, the specific responses are greatly inhibited when the VST is expressed, the CAR, compared to the VST that did not express anything. So indicating that the tonic CAR signaling actually interferes with the function of a TCR in T cells. So what I've shown you so far is that there's ample evidence that the presence of tonic CAR signaling is actually detrimental to the cells. It can produce toxicities. In extreme cases, it may lead to exhaustion and cell death. In mild portions, so to speak, it can still increase differentiation and deplete these long-lived populations before you inject the cells. And so in order to really optimize the CAR function in vivo, one has to minimize the CAR signaling, tonic CAR signaling in T cells. So how can we do that? I want to briefly review the strategies that we can use to reduce the tonic signaling in CARs. And first is obviously to modify the structure of the CAR, because this is one of the key components that actually facilitates tonic signaling. And in this case, I want to show you one example kind of firsthand. In my group, we were able to attenuate tonic CAR signaling by simply replacing a spacer in the transmembrane domain from a CD8 alpha to IgG short hinge and the CD28 transmembrane. And this simple trick actually resulted in significant attenuation of cell toxicity in vitro and boosted the in vivo function of the CAR. This is again with retroviral vectors. We see with a lentiviral vector it doesn't really have any effect because of this absence of this positive feedback loop. But for example, there are CARs in which this trick doesn't really work. In the GD2, it doesn't seem to help because the majority of tonic signaling is happening through the CFE and not through other components. Likewise, when we express this CAR in a multivirus specific T cells, we can see that the responses through the TCR are now unimpaired. The cells are able to mount the full magnitude of TCR responses to a specific peptide. So the cells essentially do not notice the CAR anymore. It doesn't seem to bother them, and they are able to fulfill their potential. So modifying CAR structure is a powerful way to reduce tonic signaling. Another approach is obviously to modify the expression level of the CAR in the system. And one example I've shown before, when the expression of the CAR can be attenuated by an iris or by other methods in your expression vector, this really leads to significantly improved anti-tumor activity in vivo and foster a good expansion of these T cells in peripheral blood. Likewise, one can use a non-LTI promoter, or another promoter that will not be activated by tonic CAR signaling. In this case, we use the EF1 alpha promoter in the context of self-inactivating lentiviral vector. And this resulted in a slightly lower expression of the CAR, but due to the absence of this positive feedback loop, the cells were much healthier and produced better anti-tumor activity in vivo. And again, you can see the expansion on the right was significantly higher than those expressing with it from a retroviral vector. Similar results were reported by the Penn group a few years ago from Carl June, in which the expression of the CD28 zeta CAR in a lentiviral vector from EF1 alpha promoter was high enough to promote tonic signaling in the cells. And in this case, they were able to further attenuate the expression by using a truncated PGK promoter, which resulted in a low CAR expression, but dramatically improved the anti-tumor function in vivo, as illustrated here. And finally, CARs can be engineered to be expressed from an endogenous locus in a chromosome, as shown here in a report last year from Michelle Sadeline's group and MSK, where they introduced the 28 zeta CAR in a TRAC locus of the DNA, and that resulted in significantly lower expression level on the cell surface, and this led to dramatically improved anti-tumor potential in vivo, and also resulted in less exhaustion of the cells when isolated from in vivo tissues. So really, this for example tells you that when it comes to CAR expression, often less is more. You do not need to drive high expression to achieve optimal function. Sometimes you actually need to dial it down. And so before I close, I wanted to briefly illustrate and introduce another alternative approach by which you can use to attenuate tonic signaling. Instead of permanently decreasing the expression level through different promoters, one can reversibly inhibit CAR expression during the expansion in vivo. And this, for example, we can use a TET-off system, as shown here. In the presence of a small molecule DOX, the CAR is not expressed. And so once we inject these cells to the patient, the CAR T cells upregulate, T cells upregulate the CAR in the absence of dogs for the first time within 3 to 4 days, as shown here, and actually go after the tumor and receive antigen-induced stimulation instead of tonic signaling through the CAR. And if needed, we can always inhibit it back by administering dogs to the patients. And so one of the biggest advantages of this system is that because there is no CAR expression during ex vivo culture, the cells look intact. Their differentiation is the same as the control cells that don't have any CAR at all, and they retain this beneficial naive-like cells compared to the cells that constitutively express the CAR from the retroviral vector. And this ultimately leads to a better activity in vivo, better expansion, and a better survival advantage, indicating that by hiding the CAR expression and deferring expression and letting the T cells express the CAR for the first time after they're infused to the patients actually prolong their activity and may boost the anti-tumor potential. And so I will stop here and quickly summarize what I've said. Tonic CAR signaling is bad for the cells. It can lead to a lot of bad things and ultimately jeopardize anti-tumor activity. And to optimize the CAR, you need to minimize the tonic CAR signaling. You can do it either by modifying the CAR structure or by modulating expression level in various expression systems. Thank you. I'll take your questions.
Thank you, Max. We have, I believe, one microphone in the center for questions. We have 5 minutes for questions. I'm going to start with the first. So you showed something very interesting, a couple of things. So all CARs dimerize, naturally dimerize based on the extracellular spacer, whether it's an FC, whether it's CD8 or CD28. And then you showed that by switching the extracellular domain by switching the hinge of the BB zeta CAR and the transmembrane to a CD28, you increase the function and presumably decrease the aggregation of tonic signaling. So what, and then I'm thinking about data that Michelle Sadline showed this morning about the induced T cells when expressing a CAR, losing CD8 alpha beta expression. What do you think about CARs and the interaction with endogenous molecules on the cell surface and how they co-opt those, how they bring those into the CAR cluster, and how that drives some of the tonic signaling?
Oh, this is an excellent question, and we don't have any solid data to illustrate it, but actually in T cells what happens is a lot of components of TCR signaling and auxiliary molecules get recruited to the lipid rafts, and I believe the same may happen when a CAR is signaling. So there's natural aggregation of all the CD8 and CD28 potentially. And so it is very likely that the CARs may interact with some of those signaling components, and this may lead to attenuation or boosting the tonic signaling in general. And this is definitely an area that we wanted to investigate in the future, but I don't have any mature data for you right now. But it's an excellent point, yes.
Okay, second question. So as we build new CARs, and as we go back to think about the Chimeric and Yescarta, and whether those molecules are optimized for function, is there a simpler way than intrinsically testing out each molecule, each single part of the molecule?
Well, there's multiple ways by which you can test the CAR. I guess the question is, what is the most revealing, what is the most predictive? And some people think it's the in vivo, and they don't even bother doing in vitro assays until they go in vivo in mice. But then again, the results that you get in vivo, do they correlate with the function? I think doing some kind of preliminary in vitro testings with looking at various aspects of the CAR signaling would help a lot, and we clearly know that the more tonic signaling we have, the less cells perform in vivo. There's multiple evidence to that, and so this is very easy thing to detect in vivo just by running Western blot, looking at spontaneous phosphorylation, or looking at activation of expression of activation markers, so on and so forth. And so this can be kind of included in the pipeline, I guess. And But ultimately, sometimes a lot of what we do now is very empiric, and stuff that works great in vitro may not work best in vivo. And so ultimate test will be in the patients, I guess.
That's difficult to do and expensive.
Yes. Versus.
Okay.
Yeah, is there any evidence that the affinity of the CAR makes a difference to tonic signaling?
Does the affinity of the CAR make a difference? Well, I think it has to have, it has to reach a certain threshold to facilitate, optimal antigen binding. And if the affinity is insufficient, I guess it may not form this immunologic synapse strong enough. But high affinity CARs are not necessarily better than low affinity CARs. And so I think there's kind of the bell-shaped curve at which having too high of affinity may introduce this hook effects or let the cells be stuck with the tumor cells.
I was actually really asking about what's the effect of that on tonic signaling.
Oh, that's, we don't see really correlation. I think what happens is the SCFEs, how SCFEs affect the tonic signaling, is that what you're asking, is that what happens is their kind of lateral mobility and ability to stick to each other, not so much to the antigen. And so the affinity to the actual antigen may not matter, although in many cases the propensity for tonic signaling is actually programmed in the CDR regions, and that's what's dictating the affinity. But we don't really see a good correlation. Sometimes low affinity CARs, tonic signaling are really like crazy, and sometimes high affinity are pristine. So at least in our hands, we don't see any dependence.
Thank you, Max.
Thank you.
Our next speaker, you can give a round of applause.